The red colors represent a small number of human immunodeficiency virus type 1 infected individuals who can naturally control the infection in the absence of antiretroviral therapy. The EC phenotype is rare and is found in less than 1% of all HIV-1 positive individuals. Researchers have long strived to use the EC phenotype as a model for immunity to resolve HIV-1 disease progression with the aimed outcome of a functional cure. In this study, we conducted separate untargeted quantitative proteomics and genome-wide transcriptomic analysis to identify unique features of EC phenotype. Furthermore, we employed untargeted proteomics and transcriptomics analysis in male EC to stratify the significance of identified pathways on a complex level. The individual omics level analysis was first performed on transcriptomic and proteomic data separately to identify features associated with the EC phenotype. Previously, we have shown a strong sex bias in the EC group and a major difference in the gene expression in male EC compared to male healthy. Therefore, in this study, we first focused analysis on male cohort. Overall, the data indicates that HIF1 signaling and metabolic pathways such as glycolysis and pentose phosphate pathway distinguish the male EC phenotype compared to other cohort at a proteomic level. Identification of biological functions are often dependent on signaling cascades and activation of targeted transcripts. Similarly, HIF signaling leads to translocation of HIF1 alpha and HIF1 beta into the nucleus and transcriptional activation of HIF1 target genes. Therefore, to better understand the cellular phenotype associated with the EC, we integrated expressed transcript information in proteomic data. The results show that increase in proteins associated with HIF signaling and glycolysis independent of HIF1 alpha levels in total PBMC are characteristics of male EC phenotype. As we observed a difference in HIF1 alpha protein levels in male and female EC, we next looked into whether altered HIF1 alpha levels may have an effect on transcriptional activation of HIF target genes. Therefore, the transcript data of a complete cohort was used to study the expression profile of HIF target genes. Our research suggests that there are sex-specific difference in activation of HIF targeted genes, and higher proportion of HIF1 alpha and HIF1 beta is localized in the nuclei of CD4 and CD8 plus T lymphocytes in male EC under normaxic conditions. An enrichment of glycolytic pathway was a feature detected in our male EC cohort, and glycolytic genes are mostly under transcriptional control of HIF1 alpha. Therefore, we evaluated intracellular metabolite levels of glucose, lactase, glutamine and glutamate with their corresponding transport surface expression of lymphocyte, monocytes, intermediate and non-classical monocytes in both the male and female EC and healthy cohort. The research shows that EC exhibit a unique metabolite uptake and secretion profile compared to healthy independent of sex. Earlier studies have shown how the mammalian target of rapamycin pathways is upregulated in EC and a major regulator of HIV-1 latency. mTOR pathway is also an upstream activator of HIF signaling and metabolite reprogramming. Therefore, we sought to evaluate the activity of proteins involved in mTOR signaling such as AKT, mTOR, S6K1, 4E-BP1 in male and female EC and healthy cohort. The research indicates that female EC have higher activation of AKT phosphorylation not seen in male EC. In conclusion, we saw that glycolytic modulation together with the altered HIF signaling contributes to the unique characteristics of male EC phenotype. Furthermore, induction of HIF signaling may result in metabolite reprogramming together with the production of antiviral proteins and modulation of immune cell functions. Together, these factors contribute to the EC phenotype of natural control of HIV-1, but how it directly affects HIV-1 persistence remains to be elucidated. However, the EC cohort may not be ideal model for functional HIV cure, given that sample cohort of both the male and female EC is limited. As a proxy for immunological control, long-term ART-treated individuals could be of interest. This group could potentially be used for identification of immune cell function during long-term suppression of HIV-1 and clarification of how the immune metabolic profile is related to HIV-1 persistence.